Good morning, everybody, and I want to again thank the organizers as well, David Hood, for the invitation to speak here and tell you. I'm going to start with a little bit of historical perspective of how we got into where we are, and then show you some newer data. And it's been really nice. Gianni set this up perfectly, so I don't have to give a lot of introduction, as did, there was a talk the first day of the meeting about what we know mainly about satellite cells, and that's regeneration. And it's clear that and we know a lot about the regulatory steps, the transcription factors, starting with the PAX7 positive satellite cell and how it undergoes activation and differentiation in regeneration. So, and they're absolutely required for that, and that's been really well established. Now, it's also clear that they participate in hypertrophy in response to resistance training, which is what we've been really interested in over the last many years. And whether they go through this same program and how this process really happens, so that if you want this fiber to grow, you need to add nuclei, or you do add nuclei, so that you can support this amount of additional amount of cytoplasm. But we started out the study about 10 years ago with the hypothesis that, therefore, if you depleted satellite cells from muscle, you couldn't hypertrophy the muscle. And the way we did that was by a genetic model using Cre-Lox technology, and that is that we obtained a mouse from Gabby Cardone in which the PAX7 promoter, which is satellite cell specific, drove Cre recombinase, crossed that to a mouse with a strong constitutive promoter, but there's a flock stop codon in front of the diphtheria toxin A gene. You cross these mice, and this diphtheria toxin A gene is not active until you treat the mouse with tamoxifen, and what that does is it effectively kills satellite cells. It's a very effective treatment, and we can deplete satellite cells greater than 90%. And so we then thought, okay, now, and I want to say that if you inject cardiotoxin into the tibialis anterior of this mouse, there is no regeneration. The muscle stays like mush. So satellite cells are really important for that. And the way we induced hypertrophy was with the synergist ablation model. And that is, we removed a small part of the gastroc and the soleus, which causes a mechanical overload of the plantaris muscle, and it hypertrophies tremendously. And we hypothesized, as I said, in that model that you would see no growth because there's very little satellite cell content remaining. And to our surprise, although we did effectively, I'm Gianna, I see how this is, I'm going to be back and forth in the wrong button here. What you can see, we did effectively eliminate myonuclear accretion. So that is that whereas normally during hypertrophy you do add myonuclei, they increase. We can see here in the black bar the tamoxifen, the satellite cell minus animals. There's no myonuclear accretion, and yet growth after 2 weeks, so SA is synergist ablation, 2 weeks, growth was unaffected. This was a huge surprise to us, and we counted myonuclei, both just DAPI-stained nuclei inside the dystrophin-stained membrane, and we also on isolated single fibers, and got the same result in both cases. So that is a very big surprise to us, and that we then showed that basically on these single fibers, the myonuclear domain just gets bigger, so that up to a point, for 2 weeks, you see tremendous growth, and with no change in myonuclear number. So there's actually a great deal of flexibility in the myonuclear domain, that the existing myonuclei can ramp up their transcription to support growth up to a point. And if you let the animals go longer, so here I'm showing you, 1 week, 2 weeks, 4 weeks, and 8 weeks. By 4 weeks, you start to see that growth is actually blunted. And here I'm showing cross-sectional area at 8 weeks, and you see that there is a limit to the growth without addition of myonuclei. And one could hypothesize that it's a myonuclear domain ceiling, which is entirely possible, and we've been trying to test to determine if that's the case, and we don't really have any evidence of that to this point. But what we do see at 8 weeks is tremendous fibrosis in the muscle in the absence of satellite cells. So what you can see by two methods, both picrocereus red staining or wheat germ agglutinin for glycosaminoglycans, that the muscle becomes very fibrotic. And what we hypothesized was that it's actually physically limiting the growth of the fibers. And so we've done a lot of work to try to understand how the absence of satellite cells might lead to fibrosis. And what we've come upon is that satellite cells not only fuse to a fiber, but they're constantly, as Gianni noted, they're really communicating with all kinds of cell types in muscle. Fibroblasts mostly to regulate the extracellular matrix, and to Gianni's talk in endothelial cells maybe, and capillaries signaling to satellite cells. We have evidence that satellite cells signal back to endothelial cells. So it's a very dynamic environment. And I'm not going to show you any of those data, and I want to encourage you to go to John McCarthy's talk, which is tomorrow morning, where he's going to focus on this intercellular communication and present our work on that tomorrow morning. What I more want to focus on is what about the role of satellite cells. So what mostly my talk based on this, synergist ablation is a very supraphysiological model. The muscle doubles in size in 2 weeks. It's not translated. It's surgical. It's non-reversible. And so we really wanted to develop a model in the lab that was more translatable, and that it made more sense and that could potentially have more relevance to human hypertrophy in response to resistance training. And also we wanted a model that was reversible, because we really wanted to study myonuclear dynamics, satellite cells, with training, and then after you discontinue training. And so what we decided on was progressive weighted rear winding power. And this was a model developed by Corey Dungan with the help of Kevin Murrock, who is here in the audience. And basically it's very simple. You just acclimate the mouse to the wheel, and then over the course of several weeks you just add 1 gram of weight. And it's only on one side of the wheel, so it's unweighted. It's unbalanced. And so the mouse runs, and as the weight increases, the momentum of the wheel, it goes really fast, and the mouse gets off. And then the wheel starts and it jumps back on. And so as the weight gets heavier, it has a harder time. It really has to struggle to get it going. And then it goes too fast and it jumps off. So all night it jumps on and off the wheel. So it's a combined endurance and resistance training model, which we really liked, thinking that that's really the most effective way to improve the health of your muscle. So that's the model that we did. And we wanted to see in this model, which we thought was more physiological, what the effect of depletion of satellite cells. So we subjected our PAC7 DTA mice to 4 and 8 weeks of progressive weighted rear winding. And what I want to show you to start is that, and the other advantage of this is that you can look at all the muscles in the animal. So you're not restricted to the plantaris as we are with our synergist ablation model. And so I'm going to show you results here from both a predominantly oxidative muscle, the soleus, and a glycolytic muscle, the plantaris. And you can see in both cases, which has been shown with running, that satellite cell content, so here's sham, 4-week, 8-week of power, you can see a nice increase in both the soleus and the plantaris in satellite cell content, which is completely eliminated following tamoxifen treatment, so in these satellite cell minus animals. And we were a bit worried when we started this that perhaps the running would, because of course we don't delete 100% of the satellite cells, nothing in biology is 100%. We thought the satellite cells might repopulate, but they don't. You can see that they stay very effectively depleted with the running. What you can also see is that this depletion of satellite cells with running or during running does not at all affect the nice fiber type shift that we see in the plantaris. So what you can see here, sham, in both the 4-week and 8-week animals, whether or not they had satellite cells, they had a nice increase in 2A fiber content, a decrease in 2X and 2B. So there was a nice oxidative shift, which was completely independent of satellite cells. We saw a trend towards more type 1 fibers and fewer type 2A fibers in the soleus, but it didn't reach significance. But again, it was not affected by satellite cell content. Now, what we also saw, as we did in our synergist ablation model, that running was associated with increased myonuclear content. Okay, so 4 weeks and 8 weeks, in both the soleus and the plantaris, there was a nice increase in myonuclear content, which was eliminated in those animals that were tamoxifen treated. So the model seemed to be working, and of course the main point is what we saw was blunted growth. So there was no satellite cell myonuclear addition, and what you can see in the soleus at 4 weeks and 8 weeks, there was nice cross-sectional area increase, which was blunted in those animals without satellite cells. And one thing I want to just point out here in the plantaris is that at 4 weeks, there was no significant yet CSA increase. And yet, as I showed you, they already had myonuclear accretion. And so, again, it spoke to us that the myonuclear domain is very flexible, because in these animals, they had already started to accumulate new myonuclei, so the myonuclear number is up, but CSA was not. So now the myonuclear domain is much smaller for a period of time until finally CSA catches up. So we thought this was very interesting, and that it's clear that growth is affected in the absence of satellite cells at both 4 and 8 weeks. So I should say that we looked, of course, at fibrosis, seeing if this was similar to the synergist ablation model. Was the muscle more fibrotic, and perhaps that's contributing to the lack of growth? And that we did not find the same results as we did with synergist ablation. There was no additional fibrosis. So we decided what we wanted to do was look to try to see what might be different about the myonuclei in the animals in which there had been satellite cell addition compared to those where there were not. So we undertook some single myonuclear or single nuclear RNA sequencing. And that is that we subjected the mice, both with and without satellite cells, to 4 weeks of power. And then we mechanically dissociated the muscle so that we not only got single cells, but also broke up the fibers, so we were able to isolate myonuclei. And we subjected them to single nuclear RNA-seq. And what we found is here, and I know this is complicated, it's a UMAP of the clustering, the graph-based, unbiased graph-based clustering with both populations combined, okay, from the soleus. And, you know, using the programs, you can discern which populations are from the mouse with or without satellite cells. And what we did here, and I want to focus your attention on the myonuclei. We have other cell types there as well. But first off, what I want to show you is that the myonuclei are incredibly heterogeneous. And this is clustered based on myosin content for the myonuclei. And so you can see here that there are 2 clusters of type 1 expressing type 1 myosin expressing myonuclei, and they differ, and I'm not going to be able to go into a great deal of detail because of time, and this is published work, but this is based on the one population of myonuclei are much higher expressing metabolic genes, and the other is more highly expressing ribosomal genes. So it's really very interesting to see how heterogeneous the different kinds of myonuclei are. But what was really most interesting to us is that only in the animals that lacked satellite cells, so had no myonuclear accretion, this new population of myonuclei appeared that we called cryptic myonuclei, which are expanded here. And what you can see is that they're expressing myosin heavy chain, so they're myonuclei, but they also were clustered very closely to the FAP cluster, so fibrogenic adipogenic progenitors. So they were now expressing some extracellular matrix, some different genes, very different than any other myonuclei that we see in normal exercised mice with satellite cells. So what we think is happening is that satellites, a lack of fusion, that somehow the existing resident myonuclei sense this, and it causes dysregulation of their transcriptome, which may contribute to this blunting of growth. This is something that we're trying to explore in more detail now. So what this the finding here, let me go back, that this finding that both lack of satellite cells, over time blunts growth, lack of satellite cells causes dysregulation of myonuclear transcription in response to power, made us think, well, you lose satellite cells as a function of age. They are decreased in abundance. And we thought, well, and as you hear all the time, and I read in grant applications, loss of satellite cells with age causes sarcopenia, right? This is what is kind of dogma. We thought, well, then this is a premature aging model. Here we've killed satellite cells. Now what we did is we depleted them at 4 months, and we let them sit in their cages for 20 months. So we treated all the mice at 4 months with tamoxifen. One cohort we killed at 5 months, so it had lived 1 month without satellite cells. And as I said, the rest we just put in their cage, sedentary, and let them age for 20 months. So we killed one cohort at 5 months and one cohort at 24 months. And one thing that we, again, this is a lot of mice and a lot of time, we're worried potentially that the satellite cells would repopulate. The fact is that they don't. So here I'm showing you that here on this bottom panel, comparing just vehicle treated, so satellite cell replete muscle, over time, comparing 5 months to 24 months, you see that you do see a decrease in satellite cell content, different for different muscles. And that in the tamoxifen-treated animals now, comparing the young versus the older ones, the satellite cells remain very effectively depleted. So these 24-month-old mice have lived 20 months of their life with very, very few satellite cells, okay? And so we thought, okay, what do the muscles look like? Has no effect on myofiber size. So this was an incredible surprise to us. So you can see that there is sarcopenia, that fiber size is decreased with age to a different extent in different muscles, but there is no effective satellite cell depletion. So the role of satellite cells in aging and in muscle homeostasis is really in question, I think. And I want to just mention that I know I saw an abstract that I'm really looking forward to see the poster today by Corey Flynn, where she and her lab have identified a population of potential stem cells, HOX11 expressing stem cells, which may be involved in just homeostasis of muscle and myonuclear turnover, because these results would suggest that if we deplete satellite cells, either myonuclei don't turn over for the life of the animal, that your myonuclei are stable for your whole life, which seems unlikely to me, or there's another population of cells that aren't really involved in muscle adaptation. I mean, they can't substitute for satellite cells in growth because we don't see myonuclear accretion. They can't substitute for satellite cells for regeneration, but maybe they're specifically involved in homeostatic maintenance of muscle throughout the lifespan. So in the final couple minutes of my talk, I want to get to this detraining. Before I do that, I wanted to put in some human data. So we just completed, I'm going to go back so that you don't get distracted. We just completed the master's clinical trial, which was a clinical trial in older adults. And the point of the trial was to look at the effects of metformin on response to resistance exercise training, with the hypothesis that would augment training outcomes and reduce heterogeneity. And so we completed the study, and we had just performed a secondary analysis to see if satellite cell content was associated. I asked Gianni about do a correlation in satellite cell content or change in satellite cell content with increase in fiber CSA in this resistance training trial, which was progressive resistance training for 14 weeks in older individuals over 65. And I'll just cut to the chase and say, sort of similar to what Gianni said, there was no correlation either in this number of satellite cells to begin with, nor the change in satellite cells with the degree of hypertrophy in these individuals, which was disappointing because that was sort of the idea, and our mouse data would suggest that might be the case. But that caused us to turn to different cells within the muscle. You know, the muscle's very heterogeneous, all kinds of other cell types. And based on our work in other projects in the mouse, we turned our attention to macrophages. Muscle has a high content of M2-like resident muscle macrophages, these alternatively activated. And what we found is doing correlations to various other features in muscle. What we found is the content of the M2 macrophages, that is those expressing CD206, which is a classic anti-inflammatory macrophage marker. You see the change in the macrophages is really nicely correlated to the increased change in CSA. And it's also correlated to the change in satellite cell content. So maybe it's not just satellite cell content, but who they're talking to. Again, talking about this communication that may change their function and allow them to contribute more effectively to hypertrophy. So now, just in the final couple minutes, I'm going to share with you, the detraining results. So the mice undergo nice hypertrophy. So what we did, Corey Dungan, together with Kevin Murrock, put these animals in the cage, let them run for 8 weeks, and then 3 months of detraining. And to look at the plantaris, what you see is that a nice training result, change in fiber type, oxidative shift, which is lost after 3 months, decrease back to 2A fibers, return of glycolytic fibers, the increase in fiber CSA, both total and fiber type specific, is lost with detraining, as are myonuclei. So what you can see is that the myonuclei that were gained, either counting on single fibers or in cross sections, are returning back to control levels, they're lost, and following 6 months, they really are back to untrained level. So that this is a really nice model. And so if it's not myonuclear number that might confer a training benefit or a memory of training, we heard a talk yesterday, and Adam, I think, referred to Kevin's work, that there are epigenetic changes. And in this mouse model, in this running model, power, we can examine that, and especially using a mouse, which allows one to specifically isolate myonuclei. So I'm not going to describe the mouse in much detail, but just say that with doxycycline treatment, the human skeletal promoter drives the expression of GFP, which is a fusion protein with H2B, so the GFP is in the chromatin, and you can specifically then label and isolate myonuclei and profile them. And what Kevin and his collaborator, Ferdinand van Walden, have done is shown, using this model, epigenetic changes which are stable with detraining. And both Ferdinand and Kevin are here in the audience, and I encourage you to talk to them about this, their epigenetic studies in the memory of training, really important stuff. So just one final minute, it's a little bit phenomenological, but I showed you results of detraining with the plantaris. We also did the soleus, and I can say it did not detrain after 6 months. It still had higher fiber CSA and higher myonuclear content. And that's maybe not surprising because the mouse is now 9 months old and it's bigger, and the soleus is a postural muscle, so it just didn't detrain, it's carrying around more weight. But looking at the mouse and using this HSA GFP mouse so that you can specifically look at myonuclei, really interesting observation that Kevin made. And that is that only in the soleus, early during running, there's signs of damage. So this is embryonic myosin, and this is not apparent in the plantaris. And if you look here, the green spots are myonuclei, and they're mostly peripheral, and that's cool. That's where they're supposed to be. The blue arrows show unlabeled nuclei in the center of fibers, which are presumably satellite cells which fused in, and most of those fibers are embryonic myosin positive. But really surprisingly, if you look here as a fiber, and there's a green nucleus in the center, and that fiber is not embryonic myosin positive. So, and here's another one with a green central myonucleus that is embryonic positive. So the dogma of what's going on in terms of adult muscle adaptation is not, perhaps we know less about it than we think we do. And we try to think that there's this easy route with regeneration, and maybe it's much more complicated. So with that, I'm going to close, and I think I've really gone through each one of these conclusions, and I hope made those points clear. The take-home message that I want you to be left with is this here at the bottom, that the satellite cell contribution to hypertrophy may be fundamentally different than to muscle regeneration and to muscle maintenance throughout the lifespan. So I think what I have come to realize, having studied satellite cells now for 30 years, is that what we don't know about satellite cells is a lot. So with that, I'm going to stop and acknowledge those who are most directly involved in this project. I mentioned Kevin, who I said is here, Corey, Davis, and I haven't updated my acknowledgement slide since the pandemic because Kevin has now left the lab and just started his own lab at University of Arkansas. Several graduate students are now off doing postdocs. And Chris Fry, who was involved in many of the early studies as a postdoc in my lab, is now an associate professor at Kentucky, and my long-term collaborators at Kentucky. And with that, I'll stop and answer any questions if I can.
Phenomenal talk. I always love seeing all of this data, so congratulations.
Thanks, Tom.
Two really quick questions. The first one, and maybe you don't have the answer to this, but the Plantaris, you're seeing the myonuclear accretion before any growth. So do you think that myonuclear accretion is necessary for the hypertrophy to happen, or do you think it's responsive, or is it a muscle-specific effect?
I think it's really muscle-specific. I think that some fiber types, I myself think that type 2 fibers, specifically like 2B, can grow quite a bit and expand the myonuclear domain, opposite, yeah, I think different fiber types can grow and then have to add a nucleus, whereas others have a more restricted and they can't grow. They're maybe at their myonuclear transcriptional max, and that's when you've got it backwards. That type 1 fibers with all the mitochondria and that they're at their max and they can't grow unless you fuse in a nucleus. And so I think it's absolutely fiber type specific. I think it's muscle type specific. I think what I'm really appreciating is how different results are depending on the muscle that you're looking at. So I worry about our human studies where we take one biopsy from the vastus. Depending on the function of the muscle, I think what you're going to learn is completely different.
Absolutely. And just a quick follow-up. So have you ever tried to isolate those remaining PAX7 cells that seem to be responsive to nothing that you've ever done?
No, there's not many of them, and we haven't. That would be a great idea. My hypothesis is that, from Sherigam, Tugbosh's work, that PAX7 high cells, those expressing a lot of PAX7, are the really bona fide stem cells. And so that's the promoter we're using to kill them. So the stem cells, the bona fide stem cells are dead, and the ones left are the PAX7 low, so they weren't expressing as much TTA. So they're not stem cells. But it would be really interesting to isolate them and see what.
It's a good project for Ferdinand.
Yeah.
Fantastic talk. I always love seeing that data. That Chris Fry paper is one of my favorites. But my question is, your compensatory overload, did you see any, you brought up the macrophages, did you see any compensatory change in inflammatory cells or the inflammatory signaling panel that may have contributed to the increased fibrosis?
Yeah, it certainly was synergist ablation. There's an early inflammatory response, infiltration of macrophages, and they may well contribute for sure. By the time you get to 8 weeks, they could have initiated it. There aren't more macrophages by that point, but there are more fibroblasts. And so that's sort of where we focused in thinking that they were the main contributor to that ultimate fibrosis.
I'm Eleanor from the University of Arkansas. Great talk, by the way.
Thank you.
So I have 2 questions. The first one, when you induce sarcopenia, and I know you put the slide up there, but I can't remember exactly. Did you have a decrease in fiber cross-sectional area in the sarcopenic group? Like a significant decrease?
Yeah, we didn't induce sarcopenia, we just let the mice age.
Well, yeah.
Yeah, and at 24 months, some muscles have more loss of cross-sectional area than others. For example, the soleus didn't really show much. Again, different muscles depending on their function. So that Plantaris has a significant drop, gastroc, pretty great drop. So that, yeah, sarcopenia was happening, but different in different muscles.
And did you do any measurements so I know you did cross-sectional area. Did you also look at just the muscle weight of them? Okay, the muscle.
And it's down too.
And it was down. Okay, thank you. And then my second question is, so you said that satellite cells are depleted because of aging. Are there, right, like as you age, satellite cells are depleted? So are there any studies out there of like the mechanisms behind the depletion of satellite cells, or have you thought about looking at that, or?
Yeah, I mean, it might be that, capillary density goes down. So satellite cells are lost. It's just probably, there's a component of less activity. So how to tease out exactly what goes on. There are a lot of people studying that, and there are different, pathways and signaling events that are different. And so I think it's certainly multifactorial. And again, it doesn't happen at the same rate in all muscles. So some muscles at this point, 24-month-old mouse, there wasn't a loss of satellite cells. And so it's very different and I think it'll depend on so many things, as does your loss of muscle in general with age.
Okay, thank you.
Yeah.
Right, last question.
Henning Wackerhage, big fan of your work.
Thank you.
A lot of my lecture slides. So my question is, satellite cells seem to respond to the volume of the muscle fiber. So this seems to imply that they must be able to measure volume or something. And do you have an idea how this is done?
No, it's interesting you say that. And just in resting muscle, the concentration of satellite cells, or the abundance of satellite cells, is higher in slow muscles, which have smaller fibers. They're just higher density than in fast muscles that have bigger fibers. So at rest, there isn't. I think it's a mechanical thing. I don't think they're necessarily sensing volume. I think there's a mechanical, the membrane of the fiber is changing, and satellite cells sense that. They get activated, and whether they fuse in or go back to quiescence, I mean, I don't know, and we're really interested in that and how a satellite cell gets signaled to fuse in or continue dividing, and also how the myonucleus, because the myonuclei sense if there's a satellite cell that's fused in or not. And they change their transcriptional output, and certainly because of all the cytoskeleton and, that there's all this mechanical signaling that I think is responsible.
Yeah, greetings from the hippo field.
Just a quick technical question. In the power model, in the DTA ablation mice, did the DTA mice run the same?
Yeah. Yeah, in this, they did for this period of time. We've also done, well, that's just running. Over time, as mice get older, they run less. But in this model, for that period of time, they run the same.
Okay, thank you.